welcome back. Don't forget to like, share and subscribe to my channel. Today we're looking at the digestive system in the organisation topic, and we're going to look at enzymes and their special shapes. Enzymes have special shapes. They use these shapes to catalyse reactions. They're very particular about catalysing these reactions, so they usually only catalyse one specific reaction. Here we can see a diagram of an enzyme, and here is the substrate. The enzyme has a very special area called the active site, which is here, and this is where the substrate fits onto and binds together. The enzyme remains unchanged and products are formed, and we can see that the products are different to the starting substrate. This theory is called the lock and key model. In the lock and key model, the fit is exact. There is another theory called the induced fit, and this is more like a glove and a hand, where the glove to allow the hand to fit in. So in this one, the active site changes the shape, where it allows the substrate to bind with a tighter fit by adjusting. The tip to remember is that the enzyme is the catalyst, and the substance that it acts on is called the substrate. Brings us to the exam questions. Question 1. Enzymes are used in the body cells. What is an enzyme? Drawing around the correct answer. This is worth 1 mark, and our options are an antibody, a catalyst and a hormone. Remember, hormones are to do with glands. Antibodies are to do with diseases. So the answer for this question is a catalyst. Question 2. What is the part on the enzyme the substrate fits into? Is it an action site, an active site or specific site? And this is worth 1 mark. So if we think back, the answer is active site. So to remember this, use the tip. For the enzyme and substrate to become active, it needs to attach onto the active site. Question 3. If amylase is the enzyme and starch the substrate, it will give simple sugars as its products. What would be the name of the type of enzymes used for lipids and the name of the products after the reaction has been catalysed? And this is worth 2 marks. If you've been watching my previous videos, you will know that we looked at enzymes in my last video. The enzymes we looked at was amylase, we looked at protease, we looked at lipases. We learnt that amylase was to do with breaking down carbohydrates, and I'm going to write CHO as abbreviation for carbohydrates, and it broke the carbohydrates down to simple sugars. For the proteases, it broke down the proteins, and it broke the proteins down into smaller products called amino acids. And the last one we looked at was lipases, and the lipases were used for lipids, and it broke the lipids down into fatty acids and glycerol. So if we go back to the question, it's telling us amylase is an enzyme, and it breaks down starch, which is a carbohydrate, and it gives simple sugars as the products. We want to know the type of enzyme used for lipids and the products that it forms. So here we have lipases, here we have lipids, and here we have the products that it forms. So our answer is lipases or lipase, and the products is fatty acid and glycerol. The linking word for today is enzyme. When you think of enzyme, you should think of a biological catalyst. You should also think of a protein. You should think of active site. You should think of substrate. You should think of fit or bind, because the substrate would fit or bind into the active site. And when you think of fit and bind, you should think of the lock and key hypothesis, or you should think of the induced fit. When we're thinking of lock and key hypothesis, we should think of an exact fit. Thank you for watching. Join me on my next video. Don't forget to like, share and subscribe to my channel.